So good morning again. I hope you guys had a good weekend and were able to, those who are from out of town were able to go around city and visit some places. So as I just communicated to all of you, uh, today we will be discussing about another important aspect which involves the BioAssays and bioefficacy models, particularly for drug discovery process and how the drug discovery is optimized. Particularly today I am going to cover this approach for optimization of pharmacology and safety profiles of the drug, which is really very important aspect. So one of the important topic related to this area where the pharmacology and toxicology, the safety profiles of the drugs can be optimized is through manipulation of some chiral switches. This is a kind of chemistry optimization. Chiral molecules, the molecules with asymmetric carbon atom. I will be just giving you some brief background about those, what the chiral drugs are, and how those chiral switches can be optimized to achieve the desired pharmacological properties and also uh, to make the drugs safer. So the main important components of chiral manipulation and pharmacology and safety profile manipulation is there are two aspects which I will be touching upon. Is the pharmacokinetic profiles and the pharmacodynamic profiles, and what are the ways to evaluate, giving you some kind of specific case study, giving you the data generated in the lab. So before I go on to the real topic, I just want to set a stage that one of the important problem in drug development has been the large number of attritions. Attritions means there are large number of drugs when they reach to clinical studies, they fail. And there was some kind of analysis that what are the reasons the drugs fail in the clinic. This was the data which was collected in up to during 1991 and 2000. I could not find much more recent data, must have been there. But this will give you some kind of feel that what are really the factors which really lead to failure of the drugs in the clinical situation. So if you look at the situation in 1999, most of the failure uh, in the clinic of the drug, they were attributed to either the pharmacokinetics or the bioavailability of the drug. Even though most of the failures, of course, they occur because you are not able to get the desired efficacy or the desired pharmacology of the drugs in the clinic. The drugs are not that effective, and that's the reason that they have to be taken out from the market. But this was really surprising that just the reasons of not having optimum pharmacokinetic properties and not having desired bioavailability of the drug can also lead to such a large number of attritions. But if you look at the situation afterwards, because this area was identified and the people started working on optimization of pharmacokinetics and bioavailability mostly during preclinical stages, and that's what the reason that they were able to reduce this to very large extent compared to what you see in 1991. Still, the large number of drugs which fail in the clinic, not the desired efficacy is there, but the toxicity still remains the problem in drug discovery, particularly the reasons for failure. Now, when you consider this kind of failures at the different phases of drug development, along the drug development phases, particularly during preclinical evaluation, phase 1, phase 2, and phase 2B. Phase 2A and phase 2B. Phase 2A is the efficacy study, and many times phase 2B is the follow-up efficacy study, generally sometime having the multicentric clinical trials. So if you look at this, most of these problems, if you are able to make sure that you optimize the safety and PK properties in early phases, you are able to get rid of most of the failures in the early phases, because if any of your drug fails in phase 2 and phase 3, it's going to be a very expensive profile. The cost is going to be very high. As you move along this pathway, the cost of drug development exponentially increases. So if you are able to phase out most of the drug during this path only and during this stage only, you can reduce the cost of drug development tremendously. And this is one of the reasons that determination of safety and pharmacokinetic and pharmacodynamic properties are important aspects, and you are able to overcome almost all the pharmacokinetic factors in the clinical situations to come. And that is what I am going to talk about, that how we can optimize the pharmacokinetics and pharmacology of the drugs in the process of drug discovery and drug development. So as I mentioned, the chiral switches in a molecule. So one of the important aspects for optimizing the pharmacology and safety of drug is through manipulation of the chiral switches in the drug. When we talk of chiral switches, these are the asymmetric carbon atoms in the structure of a molecule, which you manipulate them in either direction. Sometimes there are molecules, they have single chiral molecules, some compounds they have multiple chiral molecules, and adjustment of these chiral centers just during the process of medicinal chemistry process, you can optimize the pharmacology and safety of the drugs. Just to give you simple, those who are from chemistry, it should be very preliminary for them, but just to give you an idea what chirality is there, what we call it chirality, in the sense that when the two objects, they cannot be superimposed, then they are called chiral. When the two objects, when they can be superimposed, then they can be termed as achiral. Now, this is another way of showing the chirality with the two hands. So, the chiral center in a molecule gives rise to two different molecules, which are commonly known as either enantiomers or stereoisomers. There are different terminologies which are used when we try to define the chirality. Just to give you an importance, how important the discovery in this area was, in 2001, the Nobel Prize in Chemistry was awarded for the work in the area of chiral drugs and the chirality. Professor William Nobles and Ryoji Niori, they were given the Nobel Prize for their discovery on chirally catalyzed hydrogenation reactions. And then Professor Barry Sharpless, he shared the Nobel Prize for his discovery on chirally catalyzed oxidation reactions. Particularly when it comes to technologies for chiral synthesis or chiral chemistry, the chiral separation remains the mainstay of chiral chemistry. Like when you have two molecules together, the chiral molecules together, the mixture is mostly called as racemic, and there are technologies by which those chiral molecules can be separated. But another way is also doing right from the beginning, doing a chiral synthesis, which is sometimes more tedious, but it's also an important part. So just to go on defining the chirality and initial selectivity, give an important idea that this is one of the main features of the living world. Chiral centers are very common in biomolecules, and most of these are made up of units that have the same sense of chirality. Like most of the molecules in the system, in a biological system, they have the same configuration and sense of chirality. Like all the 21 essential amino acids, they are all enantiomers, and most carbohydrates, they have D configuration. Then what we can say is that all the essential physiological processes are homochiral. Homochiral means they say homochiral means they show 100% stereoselectivity and involve only one of the possible enantiomers of the key molecule in the physiological process. And this is the basis that why it's important for us to make sure that one of the enantiomer should be important for pharmacological properties and able to give us better safety profiles. So when it comes to drug development, particularly when say chirality in drug development, I call it as a drug development in 3 dimensions, because chiral structure of the molecule takes the idea that every molecule has a 3-dimensional structure. So when exogenous compounds are introduced in the body, the physiological process show a high degree of chiral distinction, or we can call it as a chiral discrimination. So effects of different enantiomers, like the two enantiomers of a chiral molecule, they are often markedly different, very, very different. Sometimes it's very hard to believe that just changing a configuration around an asymmetric carbon center can make such a huge difference in the marked change. Different interaction of enantiomers with chiral targets such as receptors, enzymes, transporters, and other molecules. So all these factors, they make the basis for drug development in 3 dimensions. So that is the reason that the 3-dimensional structure of the molecule or the drug or the lead molecule becomes very important for consideration. And of course, the molecular basis for enantioselective absorption, distribution, metabolism, and excretion, and also the pharmacokinetic and pharmacodynamic profile becomes also important. And this is one of the area I will be touching upon today mostly. So how chirality has been really used for drug development? What is the principal scenario of chiral drug development? That either you can go for a de novo development of an enantiomerically pure drug, like right from the beginning, you develop a drug which is enantiomerically pure. It's a single enantiomer, no mixture of 2 enantiomers, which is mostly referred as racemic mixtures. Or another way is that you can have an existing racemic drug uh, and you can try to make it a switch from an existing racemic drug to one of its isomers in pure form. And there has to be some rationale. There has to be some scientific basis for that switching. So these are the 2 basic scenarios when it comes to chiral drug development area. So how the chiral switches, they are really being used by pharma companies, particularly in the area of drug development, is that sometimes these chiral switches, they are exploited to allow the line of extension of blockbuster drug. Sometimes you might have a drug which is blockbuster, which is being sold in a high quantities and it's going out of the patent. So you are likely to lose the IPR rights on that. And that's the reason that some company, what they try to do is that they use the chiral switch to get a line of extension for the IPR so that they can reap the benefits as long as they can. This is one of the idea drug development companies or the pharma company they use. So now, today, more than 75% of the new drugs, they are introduced to the market, they are single enantiomers, which is rising from about 20% 20 years before. So this is really the remarkable advancement in the sense that companies are right from the beginning trying to make sure that if there is a possibility, then they should be, if the molecule or if the lead molecule is a chiral molecule, right from the beginning, they try to determine if there is any real difference between the two enantiomers or not, and if there is a marked difference in terms of pharmacology and safety profile, then right from the beginning, they will go for a single enantiomerically pure drug rather than the racemic mixture. Previous time, some of the reasons why the racemic drugs were introduced in the market and the clinic, because the technologies for chiral synthesis, the technologies for separation of enantiomers were not really well developed. The cost of doing that was very high. So to overcome that, many of the molecules, previous molecules, drugs which are still in the market today, they were introduced as racemic mixtures. So if you look at this list, the chiral blockbusters, so 9 out of 10 drugs which are considered as blockbuster drugs, almost in 2003 sales, they accounted for almost $48 billion. And in 9 out of 10, the active ingredient is a chiral molecule. So this also gives an important that how chirality is important in terms of drug development. This is another analysis rising from 1989 to 2000. If you look at the percentage of the drugs which were approved according to chirality character, the blue bar shows the racemic drugs, the green bar shows the single enantiomers, and the yellow bar shows achiral drugs. And if you see, gradually, even in the beginning, the contribution of the percent contribution of the single enantiomers was quite high. But now, if you look at the current scenario, if you have a chiral drug, most of the chiral drugs, they are being introduced as single enantiomers rather than the racemic drug. Of course, when you have an achiral drug, then there is no problem. But in most of these cases, these kind of drugs, the proportion of these drugs are very low. As we know that by introducing a chirality in a molecule, you can increase this selectivity, and that is one of the reasons that the people who are involved in drug discovery and development, they try to prefer to go for development of a chiral molecule rather than a chiral molecule. Sometimes the cost consideration might be there. That's the reason that people try to go for development of an achiral molecule as a drug. In terms of contribution of chiral technologies, global market, there are 3 ways these can be done. As I told you, the chiral synthesis, chiral analysis, and chiral resolution. So now, because of the advancements of technologies, the chiral synthesis has really picked up the market, and it's really is the major mainstay for when it comes to development of chiral drugs. So before I go on to the things, I would like to give you some definitions of the terminologies when it comes to chiral molecule, chiral synthesis. Just to give you an idea that when we talk of enantiomers, when we talk of isomers, when we talk of how they are really defined. So when there are 2 enantiomers or the isomers, they can be defined with different terminologies. When you go into the literature, either people will say plus enantiomer or minus enantiomer. Sometimes it's D isomer or L isomer, something like that, Then S and R configuration. So S enantiomer or R enantiomer. So basically, they are the same. They are given as a different terminologies based on the properties of the molecule. So when nomenclature and definition comes, so the chirality or stereoisomerism or enantiomerism or dissymmetry is a property of an object which is non-superimposable with this mirror image I mentioned in the beginning. Then when we say enantiomers, the 2 enantiomers are the non-superimposable mirror images forms of a chiral molecule. So that's the reason we say one enantiomer and another enantiomer. Now the chiral center, the asymmetric or stereogenic center, which is mostly carbon, but sometimes it can be a sulfur, sometimes it can be a phosphorus, and also sometimes it can be a nitrogen. But mostly the chiral center in a molecule is a carbon chiral center. Then L isomer or the lever rotatory isomer, when your molecule rotates the plane polarized light in the left-handed direction or in the minus direction, then it's either referred as a minus isomer or an L isomer. Opposite, it becomes dextrorotatory or is referred as a D isomer or a plus isomer when it rotates the plane polarized light in the right-handed direction. So these are some of the physiochemical properties of the molecule which are important. When we say racemate means either it can be shown as a DL or even plus minus. So racemate means this is an equimolar mixture of two enantiomers of a chiral molecule that is normally referred as a racemate, or sometimes people try to say it as a racemic mix. So then diastereomers are the molecules which have two or more chiral centers. If you look at into this molecule, so unlike enantiomers, the physical and chemical properties of diastereomers can differ. The chemical characterization is easy and the biological activities are different. So if you look at this molecule, D-erythrose and L-erythrose, when they are superimposable mirror images, enantiomers, then the chemical properties will not be different. But if the both chiral centers are different, then they becomes diastereomers, and it becomes their chemical property also even becomes different. So diastereomers, they will have the different chemical properties, unlike enantiomers, where chemical properties will almost be similar. If you just try to do the chemical characterization, you cannot differentiate between two enantiomers. Then stereoisomers. When we refer to stereoisomers, it's the family with diastereomers, enantiomers, and geometric isomers. So stereoisomers which have the same atom-to-atom connections but non-superimposable shapes. So stereoisomers may be diastereomers, they may be enantiomers, as I mentioned. Then stereoisomers may also be configurational stereoisomers or conformational stereoisomers. So stereoisomers are going to be different compared to when you call isomers. In terms of pharmacological properties of isomers, how they are referred. When we say Eutomer, when majority of the racemic pharmaceutical, they have one major bioactive enantiomers. In a racemic mixture, when one of the enantiomer has a major pharmacological property, its major efficacy, then that particularly the isomer or the enantiomer with the major and the main bioactive property is called Eutomer. But the opposite of Eutomer is Distomer. The other enantiomer, which is inactive or less active or toxic or can exert other desired or undesired pharmacological property, is normally referred as Distomer. So the Eutomer and Distomer, they are interrelated. Then Eudysmic ratio is the difference in the pharmacological activity between the two enantiomers of a drug. So the pharmacological difference between the Distomer and Eutomer is normally given as a Eudysmic ratio. Then when we say enantiomerically pure drug is a pharmaceutical that is available in a one specific enantiomer form. So when we call it that this drug is enantiomerically pure, that it means it has one enantiomer only. As I mentioned, one of the property of chiral is this chiral switch. Chiral switch as a selection invention. This is chiral switch can be a kind of selection invention, and selection invention is an invention that selects a group of new members from a previously known class on the basis of superior properties. And one of the examples given here is about the brufen. The ibuprofen exists in two configurations, R enantiomer and S enantiomer. This was originally invented, developed, patented, approved and marketed. So 5 years exclusivity was used. There were some side effects. The patent expired. Then with the help of chiral switch, this was converted into an enantiomerically pure drug, which is an S ibuprofen or a plus chiral drug. So that allows you to get a selection patent. This is now once you have the new chiral molecule, it's a new regulated product now. This one has a superior therapeutic properties compared to enantiomer. It's used for the same indication, and sometimes it gives you an additional advantages to go for different indications also. You get a right for 3 years exclusive use of this, and new derivative or new formulation is not a new chemical entity. So this basically, when safety issues were there, one of the issue which really led to consideration that the safety issues in drug discovery are very important right from the beginning. Why we should try to make sure that the drugs are safe, they don't have any problems in the beginning. This thalidomide tragedy is mostly referred as one of the case and this is considered as one of the darkest episodes in pharmaceutical research. And this was the case which provided a lot of lessons for drug safety and regulation. And what is this? Thalidomide, it was first marketed in 1957 for as a sedative, hypnotic. This was also claimed to cure anxiety, insomnia, gastritis and tension. But it was also used for nausea and alleviate morning sickness. So this was one of the, in terms of pharmacology, this drug was like very hit, and that was the reason that in the same year thalidomide became an over-the-counter drug in the West Germany in 1957. But once the people started using this thalidomide, the drug was sold in the West Germany. Between 5,000 to 7,000 infants they were born with cocomelia. Cocomelia means deformation of legs and the hands, and only 40% of these children were able to survive. So throughout the world, now there were 10,000 cases they were reported to infants with the cocomelia due to thalidomide. Only 50% of the people they survived, and the negative effect of thalidomide led to development of more structured drug regulations and control over the drug use and development. And that's the reason that the regulatory agencies they became more vigilant. But why I am trying to cite this case here is that thalidomide is a racemic drug. So one of the property in the thalidomide is that the individual enantiomers they can racemize due to the acidic hydrogen at the chiral center, which is the carbon of the glutamate ring bonded to thalidomide ring substituent here. So one of the property thalidomide is that the racemization process can occur in vivo. It's like a inversion and the racemization. So that may plan, so that any plan to administer a purified single enantiomers to avoid the teratogenic effect will not might likely be not successful, because what happens, even if you give one enantiomer of the drug, it can be converted into another enantiomer, and the molecule will be converted into racemic mixture. So there is a property of chiral inversion is there, and that's the reason that even if you introduce this drug in a pure enantiomerically form, you are not going to address the issue of safety of this molecule. So when we consider what are the molecular basis for enantioselective drug action, there are 4 properties of the molecules which are important here. It can be pharmacology, toxicology, pharmacokinetics, and metabolism. So enantioselective pharmacology is that there are some cases, I am trying to give you some examples of how the enantiomeric selection can be important. In some cases, the racemic drugs with one major bioactive enantiomers might be there, and majority of the racemic mixture have one major bioactive enantiomer, which is Eutomer. The other is inactive and less active, which is a distomer or toxic, and it can exert other desired undesired properties. And one of the example here is that the beta blocker S-propanol, which is 100 times more active than the another enantiomer of this molecule. Then in another case, there might be the case when the racemic drugs, they have equally bioactive enantiomers. The two enantiomers, they don't have any difference or the significant difference between the pharmacology and toxicity. Only few drugs they belong to this group, and two of the important examples here are cyclophosphamide and fluoxetine. Cyclophosphamide is used as immunosuppressive drug, and fluoxetine is used as an antidepressant molecule. Then there are some drugs which have the property of chiral inversion. The drugs which with only one eutomer, but the distomer could be transformed in the body into its bioactive antibody by chiral inversion. Now this chiral inversion may be unidirectional or it may be bidirectional. As in case of ibuprofen, S is the active eutomer and R is the distomer, which is metabolically converted to S in vivo. While as I gave you the case of the thalidomide, this has the bidirectional inversion, which is called racemization. And this drug was, as I mentioned, it was withdrawn as sedative due to teratogenic effect. But I don't know, there has been some renewed interest in drug as an immunomodulator and anti-angiogenic effect, and people are trying to address the issues related to teratogenicity of this molecule. When it comes to how the chiral molecules, they might be acting differently when they interact with the target. There are different theoretical propositions which have been given, what can be the possible molecular mechanism of their interaction with the target, and why two enantiomers, they have such a markedly different pharmacological properties. So one of the theory which has been given is the 3-point contact theory, where the target has the contact molecules, contact site, where the chiral molecule interacts with this. So if it is in the right configuration, then only it will have the right interaction with the target. But the enantiomer, which is not with the right configuration, the interaction with the target will not be there, and that is one of the reasons that particular enantiomer will be inactive. Then another theory is all the four dimensions of the chiral molecule, they interact with 4 dimensions of the target. So A, B, C, D in a chiral molecule, A dash, B dash, C dash and D dash are the centers in the target which interact with them. So this is one enantiomer which interacts with the target rightly, and that's the reason it's able to show the effect and considered as an effective enantiomer. But the enantiomer which is not in the right configuration with the target will not be able to interact properly, and that's the reason it's considered as ineffective. These are all hypothetical propositions which have been shown. Some of them, of course, have been proved by molecular modeling and computational studies, but it's very difficult to functionally identify them. Now, one of the idea where the model for the recognition of chiral drugs by a target which has been given, in one case scenario, what might happen that the target protein binds to only one enantiomer selectively from the racemic mixture. This is the racemic mixture, this is the target molecule, and only one of the enantiomer binds to the target. In another scenario, what might happen that the ligand binding pocket is able to host each enantiomer individually and bind. They have the binding site for each of these enantiomers, and that's the reason that both the enantiomers are able to bind to the target. Then third scenario might be that there may be a single binding pocket which is able to host both enantiomers simultaneously. In this case, either one will bind or another will bind. The active site or the target site is configured in such a way that either one enantiomer can bind or another enantiomer can bind. In this case, the target site is configured in such a way so that both the enantiomers can bind simultaneously. So in these cases, the pharmacological outcomes for these will be different in different case of scenarios. So after giving you this level of background, now I just want to give you how the chiral drug development can be used for optimization of pharmacology and toxicology of molecules. Since I work in the field of antiparasitic drug discovery, so there is some kind of bias you will find, but overall principles and approach and the methods will be similar irrespective of the therapeutic area you are working on, irrespective of the molecules you are dealing with. So I will give you some background about how we were able to work on some of the anti-malarial drugs for optimization of their pharmacology and toxicological properties. So chiral antimalarial drugs, it has been shown that the stereoselectivity mostly has been noticed in their pharmacodynamic and pharmacokinetic properties. So just to give you an example, these are the antimalarial drugs which have been used and which are in use: chloroquine, quinacrine, quinine, mefloquine, haloperidol, and lumefantrine. Haloperidol is not in use. Quinine is just in a limited use, but chloroquine, quinacrine, mefloquine, and lumefantrine, they are still in clinical use. But if you look at all these drugs, they have a chiral center. In case of mefloquine, there are 2 chiral centers. In case of quinine, there are 2 chiral centers. Lumefantrine also has 1 chiral center, and the haloperidol also has 1 chiral center. So what has been shown is that there is a little stereoselectivity in their effects in vitro. When you try to study these molecules, the enantiomers of these antimalarial molecules, you find not too high selectivity in terms of effect of these enantiomers. The stereoselectivity is mostly noted in the ability of the enantiomers to cause certain adverse effects. So the positive pharmacology is much different, but in terms of adverse effect, there is a significant difference. Then there was a stereoselectivity also noted in pharmacokinetic characteristics like differential plasma concentrations of enantiomers, particularly the oral absorption of these molecules was non-stereoselective. Both the enantiomers of these molecules, they showed almost similar oral bioavailability. The stereoselectivity was noted in the volume of distribution and clearance. These are the parameters which gives you idea about the pharmacokinetic property of a molecule. Then what they found is that there was a significant stereoselectivity in plasma protein binding, and also the stereoselective distribution to blood cells and other tissues. And this is one of the reasons that distribution of blood cells, because the malaria parasite grows inside the blood cell, this can be an important aspect. But one of the group of molecules I am going to give you some case study about, they are popularly known as 8-aminoquinolines. They are also a group of antimalarials. 8-aminoquinolines, they are important class of drugs which are anti-infective. They have the proven clinical use against malaria, particularly for treatment of relapsing vivax malaria. But 8-aminoquinolines, they also have the promising utility for treatment of leishmaniasis, Pneumocystis carinil pneumonia infections, and also trypanosomiasis. So the broad spectrum use of 8-aminoquinolines make them very important pharmacophore. And these primaquine currently is the only drug from this class which is being clinically used. And primaquine has been in clinic almost for more than 65 years now, but it's still used as a racemic. If you look at the primaquine structure, There is a chiral center here, so this drug can exist in two different enantiomerically pure forms. There were other analogs of ataminoquinolines which are under development. Bulaquin, which was sold in the name of ablaquin, was developed as a prodrug of primaquine by CDRI. It was introduced into market, I think somewhere in 1999 or 2000, but I have tried to follow up its clinical situation, and I don't see that this drug is really being used in the clinic now. Then there was another molecule which was developed by GlaxoSmithKline, uh, originally developed by Walter Army Institute of Research in USA. That's the reason it has the accession number WR6026. This was being developed as a acetaminophen for treatment of leishmaniasis. But if you look at the difference between this ataminoquinoline and this ataminoquinoline, the chiral center has been removed here. But there were a lot of problems regarding the toxicity of this drug, so this is no more being developed for leishmaniasis now. Then another ataminoquinoline, which is in the phase 3 stage of development now by GlaxoSmithKline Beecham and Medicine for Malaria Venture, particularly for treatment of relapsing vivax malaria. Originally, this was known as Valterid 238605, again because it was developed by Walter Army Research Institute in US and later taken up by GlaxoSmithKline. This drug again, even though has a chiral center, but the clinical trial for this drug are being done in racemic form. And this is what we want to convince that no, it's not right, it's not true. When you see, when you feel that the enantiomers of a molecule may have differential pharmacology, may have differential toxicology, then it's important that even in this class, it's important for us to go for an enantiomerically pure drug. And that's the reason we developed another analog, which is known as NPC 1161. NPC is the Natural Product Center where I work at, and 1161 is the accession number. We have done a lot of studies with this molecule, and what we found that one of the enantiomer of this drug has better pharmacology, less toxicity, and is an ideal candidate for further development. But we have completed the preclinical toxicology studies with this drug and trying to approach the FDA to get an IND approved so that we can try to do phase 1 clinical trial. But what are the limitations with this drug? What was important for us to go for an enantiomerically pure drug rather than a racemic drug? Why? Why, even though for almost 65 years, primaquine is being used as a racemic drug, but why now we want to go for an enantiomerically pure drug? I will try to make some case, give you some compelling data to make sure that we can convince, we can feel convinced that yes, if we want to develop a molecule from ataminoquinoline, it has to be an enantiomerically pure molecule. So, point of emphasis is that why we want to go for ataminoquinoline. When you are trying to put so much of resources in a class of drug on a compound, you have to give a proper justification that why you want to, rather than one of the idea is that when you want to optimize an existing pharmacophore, you are trying to work on a class of drug which is already there in the market, and you know that there are some problems with that pharmacophore, then why don't you go for new pharmacophore, discover a new drug, discover a new molecule, then you will not have all these problems and properties. But then why it is important for us to optimize the ataminoquinoline pharmacophore? Why, despite of the fact that the primaquine is in use for 65 years, and now we want to optimize that? What is the need? Is this such an important pharmacophore? Is there no alternative pharmacophore available for similar properties? Yes, that is true. In the sense that primaquine is the only approved drug that kills the key parasite stages necessary for the survival of malaria. And still, there is no other anti-malarial which have shown similar properties. And what are those properties? In case of Plasmodium falciparum, this is the mature stage 5 gametocytes. I showed you the life cycle of the malaria parasite. In malaria parasite, when the gametocytes are formed, gametocytes go through different stage of development. And the stage 5 gametocyte is the mature gametocyte. Some drugs which are in development, they have some mild effect on the early gametocyte stages, but none of the drug they kill the mature stage 5 gametocytes Plasmodium falciparum. So any drug which is able to kill the mature stage 5 Plasmodium falciparum gametocyte is a good drug to prevent the transmission. As I told you that even the people are treated with a malaria drug, they still have the gametocytes circulating in their system for few days. So if they are bitten by mosquito, they are the potential candidate to transmit malaria. And this is one of the reasons that now WHO recommends that in the areas, particularly for the purpose of malaria control and eradication, every anti-malarial therapy has to be terminated with a single dose of Primakun treatment. Why? Just to clear the residual stage 5 gametocytes from the system so that the malaria transmission can be prevented. Then another stage, in case of Plasmodium vivax and Plasmodium ovale, this is the sleeping liver stage, which I mentioned was a hypnozoid, which emerges weeks to months after the initial infection and causes the relapse. And still, there is no drug, no antimalarial drug, no antimalarial molecule in the pipeline has been shown to have the effect of killing the hypnozoid except Primakun, 8-aminoquinoline. So this is one of the reasons that we have to have. This is an important pharmacophore for us, which we don't want to lose. But now, what is really the problem? Why don't we want to go ahead, since Primakun is being used for 65 years, why it cannot be used for another 100 years, What are the problems? There are some problems. The primary safety concern with 8-aminoquinoline is the methemoglobinemia, particularly the hemolytic event, especially in the population with the deficiency of glucose 6-phosphate dehydrogenase. When there is a deficiency, the genetic deficiency of glucose 6-phosphate dehydrogenase, particularly many of you, if you are biochemists, you must have read the biochemical pathway. G6PD is an important enzyme for HMP shunt. And HMP shunt is an important supplier for NADPH in the system. And particularly in erythrocytes, HMP shunt or the G6PD is the only source for replenishment of NADPH, particularly to fight the erythrocytes for oxidant insult. Erythrocytes are always exposed to oxidative stress, oxidative pressure all the time. And especially when there are oxidant drugs which are being used, NADPH help in the regeneration of reduced glutathione. Reduced glutathione is the molecule inside RBCs, which helps in detoxification of oxidant pressure. And just for the regeneration of reduced glutathione, you need a constant supply of NADPH. And NADPH being supplied by glucose 6-phosphate dehydrogenase. So if you have the deficiency of glucose 6-phosphate dehydrogenase, your capability to regenerate NADPH will be compromised. Your capability to regenerate reduced glutathione will be compromised. And this is one of the reasons that Primakun is selectively contraindicated in the G6PD deficient population. But then why? Why we are concerned about G6PD deficient population? There are a lot of another genetic deficiencies are also there. We just avoid the use of the drug in that population. Okay, make sure that people who are deficient in G6PD don't use with them. It's very difficult for us. Why? G6PD deficiency is one of the most common enzymopathy in the world. There are more than 400 million people. They are affected with G6PD deficiency. But why it is more important? Because original discovery of D6PD deficiency came up as a primacun sensitivity. When it was discovered, there were some people, they were selectively sensitive to primacun. And they found that those people were genetically deficient in glucose 6-phosphate dehydrogenase. And that is how it was discovered. Now, the G6PD deficiency is more prevalent in the area where malaria is endemic. So, if you have to use an anti-malarial drug, you cannot really screen each and every person, particularly in the field for G6PD deficiency. So, that is the reason that you need a drug which is safer in that population also. And that's the reason this is one of the most important limitation of primacun. Then there are growing evidences they suggest that the metabolic activation of 8-aminocunolene is necessary for its pharmacology and toxicology. But the pathway for metabolism, nature of these metabolites, and the mechanism of the hemolytic toxicity and efficacy are still not very clear. This is really surprising that there is a drug which is already in use for more than 65 years, but we don't know what is really the mechanism for its action. Now, we are coming up with some kind of ideas that what are the possible mechanisms. So, again, just to stress, why 8-aminocunolene? Unmatched therapeutic utility, new avenues for research opened by recent advances, particularly by our group. Then the availability of the tools to better understand the metabolic bioactivation and mechanism of hemolytic efficacy of 8-aminocunolines. Then this was very late. Discovery of enantioselective pharmacological and toxicological profile of 8-aminocunolene. This knowledge was not there earlier. This just came up very recently. Then development of technologies for separation of enantiomers of 8-aminocunolene at the preparative stage. This was one of the limitations that why people did not want to go for use of an enantiomerically pure drug, because this is going to increase the cost of the drug development, and particularly for the malaria, you need the drugs which are affordable, which are cheap. You don't want to add something which increases the cost of the drugs, particularly in the endemic areas. But now we have developed the technology for separation of enantiomers for 8-aminocunolines at robust scale. The methods are well standardized. Then another thing was that there was an access to large database and repository of 8-aminocunolines, particularly for quantitative structure activity relationship and quantitative structure toxicology relationship evaluation. So you can develop some kind of QSAR and QSTR knowledge. Then there was a potential for development of new experimental tools and models of human G6PD deficiency to predict the hemotoxicity of 8-aminocunolene. We were able to develop some experimental tools which were not available previously, particularly for evaluation that whether the drugs in the early stage of development will be toxic to human G6PD deficient population or not. So with this one, the challenge was that, the major challenge here was that there was a necessary contribution of metabolic bioactivation. So this is one of the reasons that your in vitro systems are not going to work. If you add a drug into in vitro cell culture, you won't see any effect, because there is a metabolite which has to be generated in situ at the target site, which is important. So that particular necessary contribution of metabolic bioactivation for efficacy and toxicity of 8-aminocunolene was an important challenge for development of suitable experimental models which are predictive of the human metabolic and toxic responses. Then the reactive nature, the metabolites which are generated, which are responsible for toxicity and efficacy, they are highly reactive. Their quantities are very low. They often great challenge for phenotyping of these metabolites. So this was another challenge to see how it can be done. Then, of course, this is one of the important challenge when it comes to any of the preclinical models or something like that. This is one of the challenge which has to be addressed all the time, despite of any therapeutic area you are working on. That is the differential metabolism, pharmacokinetic and pharmacodynamic response in the animal models and the humans. It's very difficult to predict if your drug is being metabolized in animals, whether the metabolism in the humans will be in the similar metabolic pathway because the systems are different. So sometimes it becomes very difficult to predict. So this offers another significant challenge for optimization of atomic analog for efficacy and toxicity. So with this one, we develop some kind of pathway for safer 8-aminoclonal antimalarials. What was that pathway? First, it involves the in silico, in vitro, and in vivo preclinical model for G6PD deficiency and the validation of models that are predictive of potential to cause hemolytic toxicity in G6PD deficient, the human with the G6PD deficiency. Development of these models was very important. Then identify the potential reactive metabolites and understand the mechanism of hemotoxicity and efficacy of 8-aminoclonolines. And the third one was design, synthesis, and selection and enantiomeric resolution of the new and lead 8-aminoclonal analogs. And finally, the preclinical evaluation of new generation analogs as more suitable developmental candidates. So this was the project which was taken up by us sometime in 2006 or 2007. So it's almost 10 years now. We have come a long way. I will be sharing with some of this data which we have developed, and why I want to share specifically this data with you, because when you apply these tools, the case is going to be similar when you are working in other therapeutic areas, especially for optimization of pharmacological and toxicological properties of any molecules, especially the pharmacophores which are already in the clinic. So again, to emphasize the pharmacon enantiomers, there are limited studies on enantioselective pharmacology, toxicity, pharmacokinetic, and metabolism of enantiomer, pharmacon enantiomers. This can exist in two enantiomers, plus enantiomers, and minor enantiomer of pharmacon. Then these are some of the, when we started this study, this was the data which was available to us. We started with this background. It was known that the capacity of isomers and the racemic to cure infection with Plasmodium salamone in rhesus monkey was almost essentially identical. This was reported by Smith et al. In 1997. And now you can consider there was a report that the enantiomers are going to be different in 1997. Until 2007, nobody really gave much emphasis to that. People were not giving that attention. So there were almost like how many years, 10 years or so. So this, but the subacute toxicity of the isomers and the racemate for this monkey, they were qualitatively the same, but pramacin was 3 to 5 times more toxic than the D-pramacin and at least twice as toxic as the pramacin. So the acute single dose of toxicity of the isomers for the mice, they were not only qualitatively different, but that the D isomer was at least 4 times more toxic as compared to L isomer, and the relevant evaluation showed that D pramacin had a therapeutic index at least twice that of pramacin. So if this advantage carries over to men, the problem that now complicates the routine use of pramacin in G6PD deficiency might be overcome. So we may be able to overcome that limitation by we can reduce the dose of pramacin substantially. Then there was another study which was from India only, that pramacin induced, the doses of pramacin used in this study, they were very high. 5 millimolar pramacin was used, but still there was a report that it produced a significantly greater increase in the methemoglobin content in reduced glutathione level. However, the release of plasma hemoglobin was greater with the plus pramacin than minus pramacin, and the qualitative effect of the normal, heterozygous and hemizygous G6PD deficient red cell were similar, but quantitatively the effect was greatest on hemizygous deficient people than and intermediate on the heterozygous. So this was the report. There were some reports on the differential metabolism of preliminary report on the differential metabolism of the enantiomers of pramacin. When the racemic mixture was administered to rats, the majority of the residual pramacin was excreted in the urine was plus enantiomer. Then using a liver microsomal preparation, there was no selectivity, and the liver S9 fraction. S9 fraction is the fraction which is considered as the post-mitochondrial fraction, which contains mitochondria and macrosomes, uh, did show selectivity with a marked preference for the conversion of minus pramacun to carboxypramacun. Carboxypramacun is an important metabolite of this. I would touch upon that. So, the more recent in vitro metabolism study with the rat liver mitochondria fraction showed that the enantiomer of minus pharmacon was preferentially metabolized to minus. This was an original preliminary report which was later confirmed by us. So, one of the major bottleneck whenever you are working with any chiral drug and you want to go for an enantiomerically pure molecule, there has to be a method, the analytical method for quantification and the analysis of individual enantiomers has to be in place. That is the first requirement whenever you are working with any chiral drug. So, maybe I will stop here so that we can come again around 11:15, and then I will show you some of the data which we have done on development of methods for analysis of these enantiomers, how the enantiomers pharmacology and toxicology studied, and how we were able to develop some models for in vitro evaluation of these enantiomers in terms of toxicology and pharmacology. Thank you. So we will come back here at 11:15. So welcome back. Um, I gave you some kind of background about how optimization of pharmacological and safety properties are important part of drug development, and the better preclinical models you have for optimization of these properties, the less chances of dropping this project in the later clinical stages it will be. And then one of the important aspects which we are discussing today is that how the chiral centers of the molecules, they can be manipulated, or how we can reduce or optimize the pharmacology and toxicology of drugs by working on uh, different enantiomerically pure components of a chiral drug. What are the methods? Uh, uh, One of the example which I am giving you is about how we were able to work on an important anti-malarial drug class 8 aminoquinolines, especially Pramacun, which is an important drug used for treatment of relapsing vivax malaria and also for prevention of malaria transmission. So, in this process, we are at the level when I mentioned that whenever you want to optimize the pharmacology and toxicology of a chiral molecule, the most important requirement, and which is an important thing, is that you need to have the analytical methods in place for qualitative as well as quantitative identification of drug, but not only drug. If there are potential metabolites which are generated from this, if you can analyze those metabolites also in enantiomerically pure form, that can be an additional advantage. So, in case of Pramacun here, as I mentioned, that the Pramacun, the racemic Pramacun has two enantiomers, the minus R enantiomers and the plus S enantiomer. Then one of the major metabolite which is generated from Pramacun is oxidative deamination at this step with the help of subsequent actions of monoamine oxidase and uh, uh, alcohol and aldehyde dehydrogenase, and generates the metabolite which is commonly referred as carboxypharmacoin. Carboxy, this amino group is converted into carboxy, and this carboxy we know from already from the literature that the carboxypharmacoin is one of the major metabolite of pharmacoin, and this is one of the metabolite which determines the pharmacokinetic and pharmacodynamic properties of the molecule. The major metabolite also determines the how fast it is generated. It also determines that how longer the parent molecule is going to remain into the system, so that it is available for the longer period, whether it's pharmacology, toxicology, or whatever effect. So that was one of the reasons that the measurement of this carboxypharmacoin was important, particularly for this project. So we were able to get the plus pharmacoin and minus pharmacoin in enantiomerically pure form as standards. Whenever you want to develop an analytical method, you have to have the standard enantiomerically pure molecule so that you can develop an analytical method. So we were able to generate or synthesize, not synthesize, but separate the racemic mixture into enantiomerically pure pharmacoin enantiomers, have these two enantiomers in enantiomerically pure form. An additional advantage with us was that we were also able to get the minus carboxypharmacoin and plus carboxypharmacoin in enantiomerically pure form. So that gives an additional advantage and power to determine whether the generated metabolite, the carboxy metabolite which is generated, whether we can determine also that it is a minus or plus. Then the 4-methyl pharmacoin here, this is a methyl additional group here. The 4-methyl pharmacoin, particularly in these analytical method, was used as an internal standard. Whenever you are trying to develop an analytical method, particularly HPLC or LCMS method, internal standards are important for more accurate quantification of the metabolites and the parent compound. So in this case, 4-methyl pharmacoin was used as an internal standard for us. So this is the analytical method which we were able to develop. The extracted ion chromatogram and the mass spectra of the racemic pharmacoin, racemic carboxy pharmacoin, and their internal standard 4-methyl pharmacoin at this one. So what we were able to see is that in case of both pharmacoin and carboxy pharmacoin, if you inject the racemic pharmacoin, you get 2 enantiomers here, in which you can quantitate easily. Similarly, in the analytical method, you can get separated the carboxy pharmacoin enantiomers also in the analytical method. And just to confirm that these are the mass spectra for these mass spectra in LCMS, the mass spectra are the one which confirm that the molecule in that particular peak is the desired molecule or not. So this was another requirement that whenever you want to determine the pharmacology and toxicology of individual enantiomers, you have to have them in a good quantities so that you can use them for animal studies, you can use them for in vitro studies, and if possible, if it is required in the later stage, you can use them for human studies. So this was one of the fractional crystallization process which was developed by us for preparation of pharmacoin enantiomers, particularly in this one, the tartrate salt is generated, and when you do the fractional crystallization, you are able to get these two salts, the enantiomers of these two salts separated, and they lead to, once you get them, then you can get the pure enantiomers or the enantiomers in enantiomerically pure form. So this was an additional requirement whenever you want to do studies with the enantiomerically pure forms. Then third one is which was important for us is, as I mentioned, that many of the metabolites which are generated from this particular compound, they are either present in very low quantity or they are sometimes unstable, and particularly when you are trying to develop the methods in the mass spectrometry, it's very important for you to make sure that whatever peaks you are identifying in mass spectrometer, they are selective, they are not coming as a background, they are coming from your original parent drug. One of the method which is nowadays being used is this development of a stable isotope labeled compound. Radioactive compound is an important aspect of doing the pharmacokinetics and pharmacodynamics, but because of some regulatory issues, regulatory compounds are being less preferred. But one of the important aspects of developing the stable isotope labeled drug is that in case of pharmacon, we were able to make the pharmacon which is labeled at 1, 2, 3, 4, and 5, 6 places. The carbon is labeled with C13. So when you take the C13 labeled carbon and mix it to 1:1 with the unlabeled pharmacon, this is not radioactive. This is a stable isotope. So this will not be in health hazard. You don't require those kind of regulatory requirements for using the stable isotope labeled drug. Sometimes even the compounds or the drugs labeled with the stable isotopes, they're also being used for clinical trials to do some pharmacokinetic and pharmacodynamic studies. So they are very safe. Normally, the labeling of the compound with the stable isotope will not make any change in terms of their chemical property, their reactivity, their pharmacology, and their toxicity. So that's one of the reasons that the stable isotope labeled compound, they are an important tool. So once we were able to make the stable isotope label compound, you just do it, mix it equally with the unlabeled pharmacon. So now, what is the difference between the unlabeled pharmacon and this labeled pharmacon is the difference of 6 molecular weight. The labeled molecular pharmacon will have an additional molecular weight of 6 compared to unlabeled one. So what is an advantage for this? The advantage for this is that when you do the analytical method and when you analyze this molecule in mass spectrometry, whatever metabolite is generated from this molecule, because it's a mixture of 1:1 pharmacon and labeled pharmacon, whatever metabolites are generated, you will see the twin peaks there. And that whenever you see 2 peaks with a difference of molecular weight of 6 in this case, that way you can make sure that in your mass spectrometry analysis, those metabolites are being generated from your parent molecule. And this is one of the way to filter actually mass spectrometry technology is very tricky. You have to make sure that there is no background. You are identifying the real metabolites. So this is one of the way to make sure. So when you see these twin peaks, so whatever metabolites are generated, they will show the twin peaks with the difference of 6. You can train your system in such a way so that only twin peaks will be identified, and then that way you can further characterize the metabolites. So this was an additional power which we added to our technology by making the C13 label pharmacon and making use of this C13 label pharmacon for subsequent pharmacokinetic and pharmacodynamic and metabolism studies. This was an advantage. So, yes.
Is it the same method like ITRAC or it's a modified form of ITRAC? Like ITRAC is used for relative and absolute quantification of unknown peptides.
Can you repeat the question?
So this method is same like ITRAC?
Yeah, it's similar to that one only. Yeah. So now, how we were able to identify the enantioselective pharmacological profile of pharmacon enantiomers by using these tools which I mentioned just now. What we are able to show that initially in the rodent model of Plasmodium burgei infection, which is a blood schizontecidal activity, as I mentioned in the life cycle of malaria parasite, it is the blood stage parasite proliferation, which is important for the pathogenesis of the disease, and most of the antimalarials they act on this stage only. So what we found is that in the blood stage activity model of malaria infection, uh, we tested racemic primaquine plus primaquine and minus primaquine. What we found is that different doses, this is the percent suppression of parasitemia, plus primaquine was significantly more active even at the dose of 11.1 3 doses. You are able to get almost 80% suppression in parasitemia compared to around 10% suppression in parasitemia with minus primaquine. So this was an indication that yes, the pharmacological activity of these two enantiomers is different. They have different pharmacological activity. This was the first confirmation on that aspect. Then subsequently, this study was extended to another model. In this model, the activity of the compound is tested on the liver stage infection of the parasite. Compared to previous model, where the infection in the animal is generated just by injection of an infected blood. In this case, the infection in the animal is generated through mosquito bite. The parasite goes into the liver first and then comes into the circulation. So during this stage, when the parasite is infecting the liver stage, the drug treatment is given. So that's the reason this model is more popularly known as causal prophylaxis model, because in this case the drug is given before the animals are infected by mosquito, so the parasite comes into the liver. So the drug is already there. So specifically, you are trying to determine the activity of the drug on the liver stage infection of the model. And this case also, what we found is that this is the dose and this is the survival of the mice. The plus primaquine was almost 4 times more effective compared to minus primaquine here. Even though if you go to the higher doses, you start seeing some level of toxicity, but yes, pharmacologically, plus primaquine shows better efficacy compared to minus primaquine. This was another show that yes, pharmacology of these two enantiomers is certainly different compared to. So it means that puts a kind of question mark that yes, why primaquine is still being used as a racemic drug, despite of the fact that the efficacy of these two enantiomers is different. But I don't know. It's not only the efficacy which is important. It is the efficacy versus toxicity which is important. The toxic dose of the compound and the efficacious dose of the compound gives you selectivity index. So it is very important for us to determine if the efficacy of plus primaquine is higher compared to minus primaquine, is the toxicity also different? So the selectivity index will be important in case if you want to show that yes, one of the enantiomer is going to be better and safer in clinical situation. So we started doing some uh, pharmacokinetic profiles of primaquine enantiomers in different models. So these are the models which were used. In vivo pharmacokinetic study of primaquine enantiomers in mice using primaquine racemate. Actually, the time when we did this study in mice, we did not have the pure enantiomers of primaquine. So that time, this study was done with racemate. Similarly, in vivo pharmacokinetic study of rhesus monkey, in rhesus monkey, again using racemate. Then in vivo pharmacokinetic study in normal human volunteers, because still we did not have enough data to show that one of the enantiomer is safer and better compared to another enantiomer. We did not have the permission to use individual enantiomers in human, but we made use of technology for the chiral separation and analytical method, and by using the primaquine racemate, we were able to generate some data in the normal human volunteers to show that the pharmacokinetic and pharmacodynamic of pharmacon enantiomer is even different in humans. And this data was very important and helpful for us to convince FDA and tell them that yes, now there is a need for us to test two enantiomers of the drug separately and make sure which one is going to be better and the safer for clinical use. Then we also did some in vitro metabolism and kinetic study of pharmacon using primary human hepatocytes. In this case, we use the racemic pharmacon as well as pharmacon enantiomers. So this is the data on the pharmacon pharmacokinetic profile in mice with a single 30 milligram per kg dose orally. These are the four parameters which we analyze: plus pharmacon, minus pharmacon, plus pharmacon, minus carboxypharmacon, and plus carboxypharmacon. The major difference, an important difference which we found was this one. The level of plus carboxypharmacon throughout the analysis, starting from 0 to is going to be 8 hours, or if you look at the level of plus carboxypharmacon, this was very low compared to what you see the minus carboxypharmacon. The minus carboxypharmacon comes down very fast here. But another thing watch here is that the level of parent enantiomers, the plus and minus, was not much different in this case. So this was the first pharmacokinetic study of pharmacon enantiomers in mice with a single dose. This was another pharmacokinetic study of pharmacon enantiomers in rhesus monkeys. Why rhesus monkey is in the sense that particularly for vivax malaria, one of the model which is there for the vivax malaria in the animals is the Plasmodium cynomolgi in rhesus monkeys. That is the only model where the hypnozoites are formed in the liver. Otherwise, in any other model, you don't see hypnozoite formation. The liver stage activity or the causal prophylaxis assay, which I showed in mice, is just a model. You are just trying to determine the activity of the drug on the liver stage of the parasite. But real relapse model, which you see in the animal model, is the gold standard Plasmodium cynomolgi in primates in the rhesus monkeys. So that's the reason, since all those efficacy studies were being done in Plasmodium cynomolgi rhesus monkey model, we generated some pharmacokinetic data of pharmacon enantiomers in rhesus monkey. And again, you look at here, the minus carboxypharmacon is very high, and the plus carboxypharmacon is very low. So this was a confirmation that the conversion of minus pharmacon to minus carboxypharmacon is very fast, while conversion of plus pharmacon to plus carboxypharmacon is very slow. Either this metabolite is distributed in the tissues, because all this analysis is there in plasma. Either this metabolite is very fast distributed in the tissues, or the metabolism of plus pharmacon to carboxypharmacon is very slow. That is what one of the speculation. Then this is the study which we did in the normal human volunteers to determine the plasma pharmacokinetic profile of individual enantiomers of pharmacon. In normal human volunteers, they were administered with a single oral dose of 45 milligram pharmacon diphosphate. The study protocol was that it was conducted with the 7 normal adult volunteers, 1 female and 6 males with different ethnicities. The individuals were orally administered 3 tablets of pharmacon 30 minutes after the normal breakfast. This is one of the requirements, that pharmacon is always used after the normal food. Empty stomach, it creates some problems. The blood samples, they were collected at different time intervals after the administration of plasma samples, after administration, and the plasma samples they were analyzed using LCMS method, which I just mentioned, for enantiomers of primaquine and the carboxyprimaquine. And this is what which we got. It's the same data presented in two different ways, where the levels of primaquine and carboxyprimaquine here is in the normal scale, and this is here in the logarithmic scale. But if you look at here again, this graph shows all of the minus carboxyprimaquine, while the level of plus carboxyprimaquine, even the log scale, was almost negligible. You don't see any plus carboxyprimaquine, while the level of parent compound, the parent drug, the plus and minus primaquine was almost similar throughout in plasma. So it shows that the metabolism of minus primaquine to carboxyprimaquine was faster, or the plus carboxyprimaquine is distributed in tissues. But again, the intriguing question here was that if that is the case, then why the level of parent drug and the parent enantiomer is almost similar in the level? If plus primaquine is metabolized slower to carboxy, where the rest of the plus primaquine is going? One of the possibility is that the plus primaquine is being distributed into tissues very fast and higher quantities. Probably that might be one of the reason, which is just a speculation. So the key finding in this study was that essentially all 100% of the carboxyprimaquine detected is minus carboxy. Plus carboxy was several orders of low magnitude lower compared to minus. It was detected in many cases under the limit of quantification, and the results suggest a markedly more rapid metabolism of minus primaquine to the carboxy, or alternatively, plus carboxyprimaquine could be rapidly converted to another metabolite or distributed into tissues. That's the reason you are seeing lower levels. But another important thing which is to be noted here is the difference between the pharmacokinetic profile of carboxyprimaquine in humans, human, monkeys, and rats, or in mice. Sorry. If you look at the graph here, the level of carboxyprimaquine up to 24 hours is very high. It's not coming down. It's only after 24 hours starts coming down. But in case of monkey, you see the level goes down. Similarly, in case of mice, the level goes down. So it means the half-life of carboxyprimaquine in human is much, much longer compared to what you see in the animal models. And this is what is going to make difference when it's the clinical outcome is there. So there are several questions which are raised by these studies. Especially one of the intriguing question is that implication of the efficacy and safety, how this data can be really implicated in the safety and efficacy of primaquine enantiomers. Now, normally the carboxyprimaquine is regarded as inactive. Then what are the relative efficacious doses of minus and plus primaquine? That will be important to determine. And what is the relative tissue distribution of these two enantiomers? That will one of the important factor to make sure. And what are the metabolites of primaquine in humans? That has to be determined. We have done extensive study on determination of metabolites. That's altogether very complex and different subject. I won't go into much details. I will present some of the data on that, but I will move on to another aspect where we did some in vitro kinetics of metabolism of primaquine enantiomers in primary human hepatocytes. So this is the protocol. I won't go into too much details of the protocol, but look at the data here. This is when we use the racemic primaquine as the substrate. In vitro metabolism of racemic primaquine by freshly isolated primary human hepatocytes. This was how the parent compound is depleted here. This is the total primaquine, this is the plus primaquine, and this is the minus primaquine. So this confirms one of the observation that the metabolism of minus pharmacone is faster compared to metabolism of plus pharmacone here. Now, consequently, if you see the formation of carboxypharmacone here compared to this one, again, the formation of minus carboxypharmacone was much faster and higher compared to what you see with the plus carboxypharmacone. So, one observation is confirmed here is that human hepatocytes, they metabolize minus carboxypharmacone faster compared to plus carboxypharmacone, and that is one of the reasons that you see higher level of carboxypharmacone compared to higher level of minus carboxypharmacone compared to plus carboxypharmacone. But one thing is very important here. This pathway of metabolism of pharmacone is not dependent on cytochrome P450s. The metabolism of pharmacone for this metabolite occurs through monoamine oxidase with the subsequent action of aldehyde dehydrogenase. The monoamine oxidase generates aldehyde, which is subsequently converted to carboxy. And what we found is that when enantiomers of pharmacone, they were incubated in vitro with a pure enzyme, monoamine oxidase A. What we found is that majority of the pharmacone was metabolized by MAO A compared to MAO B. And what we found is that again by MAO, the metabolism of minus carboxypharmacone was faster compared to plus carboxypharmacone. But then again I mentioned that this is not a CYP mediated pathway. Particularly in most of the drug discovery processes, the cytochrome P450 mediated pathways are comparatively more important. And what we found is that the metabolites which are responsible for toxicity and efficacy of pharmacone, they are primarily generated by cytochrome P450s. I will show you some of the data. It's a cytochrome P450 2D6, which is responsible for metabolism of pharmacone, bioactivation of pharmacone. As I mentioned that metabolic bioactivation of pharmacone is an important step. The parent compound is not the one pharmacologically active. Parent drug is not the one which shows toxicity. It is the metabolite which has the pharmacological activity. It is the metabolite which has toxicity. And those metabolites are generated by cytochrome P450 reaction. And what we found is that in those cases, plus pharmacone was a better substrate compared to minus pharmacone. Now that is the reason that the efficacy of plus pharmacone was higher compared to minus pharmacone. Now we are trying to pinpoint, identify the key metabolites which are responsible for pharmacology or efficacy, identify the key metabolites which are responsible for toxicity. If we are able to identify these two pathways separately, that will give us a leverage to develop something where we are able to generate higher level of active metabolites, lower level of toxic metabolites, and that will give us the idea that how to increase the efficacy of pharmacone or 8-aminoquinoline, how to decrease their toxicity. This is one of an important knowledge which we have generated during the course of this study. Now some of these data, when we did use the enantiomerically pure compounds, which is plus pharmacone only, again in this case if you see the depletion, but now main thing is that plus pharmacone is depleted here by the hepatocytes. There is a formation of carboxypharmacone, but all of the carboxypharmacone is not accounted by amount of plus pharmacone which is depleted here. It means there are other metabolites also which are generated through other pathways which account for this depletion when you are using enantiomerically pure pharmacon here. Again, in this case, the minus pharmacon, you see, this is the depletion of minus pharmacon, and this is the appearance of carboxypharmacon. This is much faster compared to plus pharmacon, as we observed when we use the racemate. But again, all of the carboxypharmacon which is generated here does not account for the all of the carboxypharmacon depleted here. So now we are in the process of identifying and phenotyping those other metabolites which might be important for explaining the pharmacology and toxicology of carboxypharmacon. So we kind of did some kind of analysis of the pharmacon metabolite which is not accounted by carboxypharmacon. And what we found is that the pharmacon which is not accounted by carboxy is little higher in case of minus pharmacon compared to plus pharmacon here. And again, the pharmacon metabolite which is not accounted by carboxypharmacon is little higher in case of minus pharmacon compared to plus pharmacon here. So the conclusion is that the in vitro kinetics of metabolism of pharmacon in human hepatocytes compare well with the human pharmacokinetic results. So this can be a good, important, nice model. During first 4 hours of incubation, the level of pharmacon depleted not accounted by carboxypharmacon was higher for minus compared to plus. So now we have extended this study to study the metabolism of pharmacon by using the hepatocytes which are generated from the induced stem cells. Those induced stem cell hepatocytes generated hepatocytes will be much better model. We are trying to do some kind of metabolomic and proteomic studies to determine what are the kind of proteomic changes which are produced by different enantiomers in those differentiated stem cells by pharmacon enantiomers, and how can we explain those metabolomic changes and the proteomic changes to explain the pharmacology and toxicology of pharmacon. But these are some of the studies, as I mentioned, we did with the 2D6 and 8 aminoquinolines. What we found is that differential 2D6 metabolism alters the antimalarial efficacy and pharmacokinetics of pharmacon in tefenoquin. I am not presenting this data. I probably might discuss the data when I discuss the animal models. This particularly study was done in the animal models where the 2D cluster was knocked out, and what we found that the metabolism of pharmacon in those animals where the 2D cluster was knocked out was different compared to the wild type mice. But then another study which we did with the pharmacon and tefenoquin and found that the metabolism from 2D6 is essential for antimalarial activity. In this case also, what we did is that we used the normal wild type animals and also 2D knockout and 2D6 knock-in animals to determine the antimalarial efficacy of these compounds. And what we found is that the antimalarial efficacy of pharmacon was compromised in the 2D knockout animals. But the main thing is that when you do the knock-in with the 2D6, you don't completely recover the antimalarial efficacy. So there are some additional factors are involved. I might discuss some of this data when I show you the animal model, particularly for drug discovery. So subsequently to this, when we were doing this study, there was a report which also appeared from the same group from the Walter Arab Army Research Institute, where they found that there was a pharmacon failure in one of the individuals with the 2D6 genotype, particularly for Plasmodium vivax malaria. So what we did is that we did some binding interactions of pharmacon enantiomers with human 2D6 and try to find out that how the enantiomers they interact with the 2D6 differentially. Is this responsible for pharmacology and toxicology of these enantiomers? I am trying to give you these examples because when you study with other therapeutic areas, the path and the approaches and the principles are going to be the similar whatever therapeutic area you study with. Whenever you find that your lead molecule is being metabolized through a particular cytochrome P450, it's the major metabolites being generated from there, then you try to study the interaction of those. When you find that your enantiomers of the particular lead drug, they are differentially interacting with the cytochrome P450, then you try to do some binding interactions. So the principles, the approaches are going to be overall similar in this case also, and when you are generally dealing with any other therapeutic area or any other molecule which shows differential interaction with the target cytochrome P450s. So this is one of the concentration and time dependent metabolism of pramacin and its enantiomers with 2D6. And again, with, as I mentioned here, what we found is that when you use 2D6, the metabolism of plus pramacin is faster compared to minus pramacin. This is the racemic pramacin, total pramacin, this is the plus pramacin, and this is the minus pramacin. So this is the amount of substrate utilized, and this is the amount of substrate depleted. Both are the similar in that the depletion of plus pramacin was much faster compared to minus pramacin. So this is different compared to what you see with the human hepatocytes, where minus pramacin is metabolized faster compared to plus pramacin. That is the overall metabolism. That includes the other pathways also. But when it comes to metabolism through cytochrome P450 pathway, particularly with cytochrome P450 2D6, it is the plus pramacin which is metabolized faster. Then there were key metabolites which we studied how they are generated. As we know that there are some phenolic hydroxy metabolites which are responsible for toxicity and efficacy of pramacin, now we know. So there were 4 metabolites, the 2-hydroxypramacin, 3-hydroxypramacin, 4-hydroxypramacin, and 5-hydroxypramacin. And what we found is that compared to other 3 metabolites, the 2-hydroxy, 3-hydroxy, and 4-hydroxy, which are comparatively less toxic, comparatively less reactive, the 5-hydroxy metabolite generation was higher with plus pramacin. We know that this is the 5-hydroxypramacin is the metabolite which we have tried to link with efficacy of the pharmacology of pramacin. So this way, with this help of approach, we were able to show that what may be one of the possible reasons that the plus pramacin has the better pharmacological activity compared to minus pramacin. This is the pathway. We identified several metabolites of pramacin generated from 2D6, but these are the metabolites which are generated in higher quantities with plus pramacin compared to minus pramacin. So I just don't want to go into too much details of this, but this is another study which we did. Then this study was further extended with the molecular docking studies of plus pramacin and minus pramacin with 2D6, and what we found is that the binding of S pramacin was higher compared to R pramacin with 2D6. Then the last lap of today's presentation I am going to discuss is, as I mentioned, that one of the problem with the 8-aminoquinoline is hemolytic toxicity or the methemoglobinemia in G6PD deficient people. So one of the assay which was important for us to develop, that how can we determine the toxicity of pramacin or the metabolite in the preclinical study. So just to study that, we did one study where we developed the metabolism linked in vitro assay for prediction of hemolytic potential of 8-aminoquinolines. So I call it as a pathway for hemolytic toxicity of 8-aminoquinoline in G6PD deficient human erythrocytes. This is just one of the presumptions that this is the pramacin with the help of cytochrome P450. There is a radical metabolite generated, and that may be responsible for toxicity toxicity of pharmacon, and we know that is the 5-hydroxy. This is the 5 position here. This is the radical metabolite here. So we know that that is the 5-hydroxy metabolite which is responsible for that. Then there is another metabolite which normally used as a or test presumed as a deactivated metabolite or the pathway of deactivation. We know that it is mediated by amine oxidases. It's a non-CYP mediated pathway. We have also shown that semi-carbonate sensitive amine oxidases beside monoamine oxidases are also involved in that. But now, normally this metabolite was considered as a safe, inactive metabolite and responsible for less half-life. One of the problem with the pramocin was it was metabolized fast to this month. That's the reason that the drug has to be administered for 14 days for the treatment. But in the subsequent next generation immunoglobulin, this problem has been addressed. But what we have found is that now even this carboxy metabolite, which is being generated, can be further acted by cytochrome P450s and converted into radical metabolite and may cause some toxicity. And as I mentioned that you saw the pharmacokinetic profile of carboxypyrmacin in mice, in monkeys, and humans. The carboxypyrmacin in humans was very high up to 24 hours. It's in the circulation, so it's going on. One of the observations in case of pramacin toxicity is that the toxicity appears late, after 2 days or 3 days after the treatment. And now we know that probably since carboxy metabolite is circulating in the system for the longer period, that may be amenable to accessible to cytochrome P450s, converted to other metabolites, and slowly slow generation of those metabolites might be the culprit in generating the toxic metabolites and responsible for hemolytic toxicity in G6PD deficient people. So that is another set of data I will not share today, but what I would like to show you is that what was the principle for developing this assay. So as I mentioned, parent drug in presence of cytochrome P450 is converted to toxic metabolite, and this toxic metabolite, particularly when it's a phenolic metabolite or it's a hydroxylated metabolite, has an important property to go through the redox cycling. It's a spontaneous redox cycling. That's the reason that even if a small amount of metabolite is generated, it has the potential to go through the redox cycling. And during this redox cycling, there is a radical metabolite generated. And radical metabolite generated. So this is what happens. That this redox cycling acts with the ferric pairs of hemoglobin, which is converted into ferric. And that's the reason that you see the accumulation of methemoglobin. And during this process, NADPH is utilized for the purpose of redox cycling. In this process, there is a free radicals are generated, oxygen free radicals, which in normal circumstances can be detoxified by peroxide dismutase and subsequently by catalase. This process requires glutathione, reduced glutathione, and that's the reason reduced glutathione requires NADPH. So if RBCs are deficient in G6PD, then this pathway will be affected, and detoxification of this radical oxygen will be affected, and that's the reason that in case of G6PD deficiency, you see higher toxicity of the drug compared to normal humans. Then there is another one where this ferric radical generated during the methemoglobin formation can be converted into ferrite radical. It acts with the radical metabolite and causes the membrane damage on RBCs, and there is a formation of hemoglobin cysteine disulfide adduct formation is there. And what happens once the RBCs are affected by this pathway, they can be phagocytized by macrophages and they can be removed from the circulation faster. This is one of the mechanisms which has been proposed for hemolytic toxicity of pramacin, but this mechanism will hold true with any other oxidant drugs also, because there are several drugs in the market which have been shown to have hemolytic toxicity, especially they are contraindicated in G6PD deficient people. So the similar kind of mechanism will hold true for those drugs too. But then what happens? During formation of ferrite radical, that acts with the hemoglobin. And the hemoglobin is denatured sometime. And the denatured hemoglobin is accumulated inside the RBCs as Heinz bodies. So the formation of Heinz bodies inside the RBCs has also been taken as a marker for hemolytic toxicity. And a lot of people, they try to do earlier studies. Heinz body formation used to be used as a diagnostic marker for hemolytic toxicity. But then there is another pathway when there is a formation of this hydroxyl radical. It has been shown to interact with the RBCs and generate a process which is similar to apoptosis in normal cells. There is no mitochondria, there is no nucleus in RBCs. So the machinery, the complete machinery related to apoptosis is not present, but there are proteases are there, the annexin binding occurs there, so calcium accumulation occurs there. So the process which is similar to apoptosis, which has been shown to occur in RBCs also, and this has been commonly named as eryptosis. So there is a possibility that these metabolites can also generate the, the hemolytic toxicity by the process of generating eryptosis. So with this one, there are some biochemical markers for hemolytic toxicity. One is, as I mentioned, the oxidation of hemoglobin and generation of methemoglobin. Another one is generation of oxidative stress, the constant generation of oxidative free radicals. Then third one is, as there is an oxidative stress, the thiols, especially the glutathione, will be depleted. So the formation of methemoglobin, generation of oxidative stress, and the depletion of glutathione, these three can be used as collective biochemical markers for hemolytic toxicity, and they can be then there. There may be another marker also like formation of Heinz bodies, process of eryptosis, then formation of hemoglobin disulfide adduct, removal of damaged erythrocyte by splenic macrophages, then similarly accumulation of hydrogen peroxide, depletion of NADPH, formation of drug radical, osmotic fragility. These can also be monitored, but these parameters, the measurement of three parameters is very easy, very simple. The process and the methods are very simple. So what we did is that we developed an assay based on these three parameters. This assay is very simple. As I mentioned that it's not the parent drug which is responsible for toxicity. It is the metabolite which is generated is responsible for toxicity. So if you add a pharmacon to the RBCs, you don't see much or significant effect. You see the effect only at very high concentrations. So we developed an assay which we call it linked in vitro toxicity assay. This is a very simple assay. This contains the reaction mixture with the buffer. You can add the microsomes, you can add the recombinant cytochrome for 52 days. NADP is regenerating system composed of NADP, phosphate, dehydrogenase, glucose phosphate. Then you add the human erythrocytes, simply incubate water bath. Principle is that in this one, the test compound and cytochrome P450, when they come together, they generate a toxic metabolite, and toxic metabolite is generated in situ, in the reaction mixture only. So even if the metabolites are unstable, they are in low quantities, they are there, they are generated in the reaction mixture only. When they react with the RBC, they give the response, which can be measured in terms of three parameters. And measurement of three parameters is very simple. You can measure methemoglobin with a coximeter, oxidative stress with a simple fluorometric DCF assay. You can measure the reduced and total glutathione using one of this kit, Promega GS Glow glutathione assay kit. Now we have changed this to LCMS method because that is more appropriate for us. Large number of samples can be put together and analyzed simultaneously. You are the use the microscope. But these three assays can be used for measurement of hemolytic toxicity response in RBC. This is one of the data which we initially generated here. This is when you use the control RBCs. The RBC is incubated with the human liver microsome, and the RBC is incubated with the mouse pooled mouse liver microsomes. So you don't see much significant response in terms of methemoglobin formation. when the primacone is there, when the primacone alone is there, you don't see methemoglobin formation. But the moment you add human liver microsomes to the reaction mixture, you see a robust response, which is higher when you use mouse liver microsomes. So this was with the primacone. We use that with other drugs also, NPC 1161A, NPC 1161B. These are the two enantiomers of NPC molecule which I showed in one of my earlier slides. Then tefenoquine, which is under the clinical development. Depsone, which is also known to produce the hemolytic toxicity similar to primacone. In all these cases, you don't see much response without microsomes. The moment you add the microsomes to the reaction mixture, you see the response. So this was this was a positive control. When you add hydroxylamine without anything, you see the robust response here. So this is without anything, without any microsomes. This was kind of a positive control. So this was the very first data which we generated with this assay, and what we found that this assay is going to work, particularly the metabolites generated. If they have hemolytic toxicity, you will be able to pick up with them. So this was assay was standardized. This assay shows 100% selectivity. We tested for clinically reported hemolytic drugs, all of them, the clinically reported non-hemolytic drugs. In this assay, we tested clinically reported 9 hemolytic drugs. Tested 8 tested positive. One of the drugs did not come up in this assay. The acetylide, some issues related to that, some metabolism issues. But one of the important thing was that in this case, we can use both normal and G6PD deficient human erythrocytes to do this assay. Because what we feel like that the formation of methemoglobin, generation of oxidative stress, and depletion of glutathione, they are early biomarkers. Early biomarkers means they will show the change in normal as well as G6PD deficient RBC. Because the G6PD deficient RBC higher susceptibility to be the late biomarkers which damage the erythrocytes, they remove the erythrocytes from the system, be more prominent in G6PD deficient RBCs. So one of the good things is that in this case, you really don't need the G6PD deficient RBCs. The response is going to be similar in normal and G6PD in this. So this was the advantage of this assay, that at this stage you don't really require G6PD deficient RBCs, but once you find any compound hemolytic in this assay, that can be confirmed using some biomarkers which we have using erythrotic biomarkers, phagocytosis assay, and what we found that they are, and especially the depletion of glutathione is more prominent in G6PD deficient RBCs. But as far as methemoglobin formation is concerned, oxidative stress is concerned, initially they are generated similar in normal and G6PD deficient RBCs. So another application of this assay which we did is that we did for CYP profiling. We just wanted to test what are the which are responsible for generating the toxicity. So in case of primacone, what we found is that there were 5 cytochrome P450s, 1A2, 2D6, 3A4, 2B6, and 2E1. They were generating high toxicity. The 2D6 was generating highest level of toxicity. This was one of the very first data which we generated with 2D6. All of the 2D6 data came from this lead only. Then in case of NPC 1161A, there were 4 cytochrome P450 involved. In case of tefenoquine, again 5. In case of NPC 1161B, which is the candidate which we are trying to propose, is an enantiomerically pure form of ataminoquinoline, which has higher efficacy, less toxicity. What we found is that the toxicity of this was being mediated only by major mediator was P450A4. So that is one of the selectivity which you get here. In this case, we tested the pharmacological, particularly in this in vitro assay. The enantiomers did not show much difference, but we were able to see the difference between enantiomers and 1161A and B. So we were able to show that for the 2C19, which is responsible for toxicity, hemolytic toxicity, and clinically, there were some reports where people have shown that cimetidine, which is the inhibitor of 2C9, has been shown to reverse the hemolytic response of dextrone. So this was another confirmation and application of this assay. So when we found that in case of 1161, it was 3A4, which was responsible for generating major toxicity, we tested some inhibitors of cytochrome P450 3A4, and what we found that if you look at the ketoconazole did not show much effect on the toxicity of Primaquine, mild effect on toxicity of 1161, more prominent decrease in the toxicity of 1161B. Similarly, another cytochrome P450 3A4 inhibitor did not have much effect on toxicity of Primaquine, less effect on toxicity of 1161A. The higher levels, the toxicity of 1161B was completely suppressed with the third 3A4 inhibitor, which is the trolinomycin. Again, in this case, mild effect on Primaquine, less effect on 1161, but toxicity of 1161B was completely suppressed. We did another study where what we did is that we took human liver microsomes, individual human liver microsomes. In individual human liver microsomes, we obtained the batches where the level of cytochrome P450 3A4 were different. So we divided into the batches where we did a study between the correlation between the content of cytochrome P450 3A4 in the microsomes and the methemoglobin response generated by the microsomes. And again, for 1161A or B, what we found that there was a direct correlation between the level of cytochrome P450 3A4 in the microsomes and the hemolytic response. So this was final confirmation that yes, 3A4 is the one must be responsible for hemolytic toxicity of this one. Then we obtained one in vivo confirmation in dog model, where if you give the ketoconazole here, without ketoconazole, this is the hemolytic response. But the moment you give the ketoconazole here, the hemolytic response can be suppressed. So this was a kind of in vivo confirmation that yes, it is the 3A4 which can be suppressed. So that is one of the ideas is wanted to float, that if whatever low toxicity of 1161B is there, if it can be used with any drug which are inhibitors of cytochrome P450 3A4, the hemolytic toxicity of this drug can be mitigated. Particularly, there are some anti-malarial drugs which are shown to have some mild 3A4 inhibition. If this drug can be combined with those, this drug will be the whatever partial low hemolytic toxicity of this drug that can be mitigated. So then, just to go further, that what is the mechanism for metabolismic hemolytic toxicity of Primaquine, we just wanted to interrogate the erythrotic pathway, and what we found is that when you are incubating RBCs with human liver microsomes and Primaquine, you don't see much erythrotic response in RBCs. This is the positive control with anomycin. Similarly, you don't see much effect on the increase in the calcium level and annexin binding with the Primaquine in presence of human liver microsomes. So at this time, what we concluded that the hemolytic mechanism of Primaquine is not going through erythrotic pathway. There are some other mechanisms which might be involved in there. Maybe I can skip this and go through immediately quickly. Another aspect which we did is that what we found is that one of the enzyme which is present in RBC, which is NRH quinone reductase 2, which protects against the hemolytic toxicity induced by Primaquine in G6PD deficient erythrocytes. And this data was generated with the help of two inhibitors, melatonin and resveratrol. Melatonin and resveratrol, which are the known inhibitors for quinone oxidase reductase 2. When we include these two in the reaction mixtures, what we found that the hemolytic toxicity of Primaquine is induced. So now we are trying to study whether how the role of malate quinone oxidase reductase might be there. Then the last one, I just wanted to see that how we can develop the agents which can mitigate the toxicity of primaquine. So we took the agents which can replenish the reduced glutathione levels in the erythrocytes, protect the cells from oxidative damage, may attenuate the hemolytic effect of primaquine and the 8-MeO quinolines in glucose 6-phosphate dehydrogenase deficient erythrocytes. And such agents may have clinical utility to be used in combination with primaquine to mitigate the hemolytic toxicity. This is a kind of approach that when you see any toxicity, any drug, there are two ways to come up with the control of toxicity. Either you can develop the combination agents which can mitigate the toxicity, or you can modify the molecule to mitigate the toxicity. This was one of the approach where you try to develop some agents, identify some agents which can mitigate the toxicity. So one of the agents which we tested is the N-acetylcysteine. The clinical studies in healthy subjects have shown an increase in the intracellular reduced glutathione with NAC. There is a decrease in oxidative stress, and there is an increase in the erythropoietin production after administration of NAC. So with that one, we tested whether the NAC can be used as a combination agent to reduce the toxicity of primaquine. So in this case, we use 5-hydroxyprimaquine as the metabolite. That I mentioned that this is the metabolite which is responsible for toxicity. And what we found is that if you include the N-acetylcysteine, the toxicity of primaquine metabolite can be greatly reduced here. Similarly, this is the measured in terms of oxidative stress and in terms of replenishment of the glutathione content. This is the 5-hydroxyprimaquine, you see depletion of glutathione, and when you add the NAC, you are able to replenish the glutathione level. So we can say that we have accumulated a large body of evidences to show that the plus and minus primaquine are not equivalent. Now we know that plus primaquine and minus primaquine are different. They are not similar. In rodent model, sporulate model, PQ was four more effective than minus PQ, but also it was more toxic. But hematological toxicity in this model is not very prominent. This is another study which I did not present the data. Probably I will show when I discuss the animal model. We use the humanized skid mouse model, which was engrafted with the GCP deficient human RBCs. What we found that the toxicity of plus primaquine was more compared to minus primaquine. In minus primaquine was well tolerated, while was showed hematological toxicity. In primates, it is clear from the literature and from our study that the two enantiomers show similar efficacy, but the toxicities are different. Then there is a definite difference in pK of two enantiomers, with minus being preferentially converted to carboxy, so that the level of minus carboxy are 10 times higher. With regard to metabolism of enantiomer, in every species, minus enantiomer is more readily converted to carboxy than plus. The fraction of the drug unaccounted for primaquine and carboxy appears quite different for the two enantiomers. Then in primary human hepatocytes, minus primaquine is much more rapidly converted to carboxy. Initial single dose PK study in normal human volunteers show that virtually all of the circulating carboxy primaquine is coming from minus primaquine, although plasma levels of plus and minus primaquine were not different. Then where to go? Primaquine is available as a racemic drug. All of line of evidences which we have generated point to the likelihood that the two enantiomers will have different metabolism, pharmacokinetics and pharmacodynamics in man, and thus they will have different pharmacological toxicological property. But which enantiomer will be better? We don't know. It depends on overall pharmacokinetic and pharmacodynamics. So the primate data suggests that therapeutic index in monkey is not equivalent for two. However, it's not known whether the therapeutic index with respect to hemolytic potential in man will be substantially improved. So the question is that we can answer this question only in clinical study. Now we have an IND in place where we plan to compare the plus primaquine and minus primaquine for safety, pharmacokinetic and normal and G6PD human volunteers. So we have already initiated this process, and probably we will be doing first clinical trial sometime in September. So these studies will set the stage for clinical efficacy evaluation of safer enantiomers, plasmodium bioextraction, both GCSPD and deficient individuals. So I may be biased for this one, but I just wanted to show you the actual data, actual practice, how we go ahead. Overall, as I mentioned repeatedly, that the principles and the assay approach are going to be almost similar when you are dealing with other therapeutic areas also. So that's what that how you can use all these models of pharmacokinetics, pharmacodynamics, and bioassay for optimizing the pharmacological properties of the drugs and for optimizing or reducing and making sure that you are able to go ahead the safer molecules in clinical trials. So with this one, I'll stop with this one that this is the molecule which we have, NPC1161. Now, NPC1161A is the plus enantiomer and B is the minus enantiomer. In this case, we definitely know that this one has the reduced toxicity and better efficacy compared to this. We have all the preclinical data to prove that. We are waiting for some kind of agency to support us for doing some clinical study with this molecule. But whatever studies which we plan to do with primaquine, probably that will give us some kind of more idea and experience to deal with this molecule. So once we know that such a material command, we also had some funding from NIH in some of the projects. There was some funding came from MMB also. It was one of the partial projects. And then we had a grand challenge award from the Bill Gates and Melinda Foundation for doing one of the study. That's from where we got some lead on the human study. So thank you very much. And if there is any question, I'll be pleased to answer. So just, I'm sure you will excuse me for being too much, teaching you too much of malaria and malaria drug discovery. But since that is the area which we have worked on, and that is, I just wanted to get you feel of actual and real data rather than giving you only the theoretical aspects and those kind of models. When you see the real data, then you feel that how really these assays and models, they really work in place. Of course, there will be some minor tweaks, minor changes when you are dealing with different kind of molecules, but the principles and practice and the approaches are going to be similar. So when everybody goes from here, most of you are going to be trained as trained malaria drug development guys, you know. Okay, any questions? No questions? Okay, in that case, if you have any question, no problem. Whenever it comes to your mind, just shoot me an email. I'll be pleased to answer, or maybe we can discuss that in-house with everybody. Thank you very much for being patient.